Om Shri Sai Ram. Welcome back for the post-lunch session of day 2 of the National Conference in Mathematical Biology, Recent Trends, Theory, Methods and Applications. We'll start with a short prayer. Om Sai Ishwaraya Vidmahe Satya Devaya Dhimahi Tanna Sarva Prachodayat. Om Shanti Shanti Shanti. Jai Bala Bhagavan Satya Baba Ji ki Jai. So, to take the session further, I invite the second MSc brothers to take the stage. Thank you.
Om Shri Sai Ram, offering my humble pranams at Swami's lotus feet, respected dignitaries, speakers and all the participants, a loving Sai Ram to each one of you. It gives me immense pleasure to introduce the speaker for this afternoon, Dr. P. Muthu. P. Muthu is an associate professor at NIT Warangal, holding a PhD in mathematics from IIT Kanpur. With extensive research and teaching experience, he specializes in fluid mechanics, numerical methods and biomechanics. Muthu has held academic positions, taught diverse courses, reviewed journals, and organized workshops. Sir has contributed to program activities, fulfilled administrative roles, chaired conference sessions, mentored students, and participated in research projects. Sir's research papers cover peristaltic motion, micropolar fluid flow, and mathematical models of fluid flow. His interests span fluid dynamics, biomechanics, numerical analysis, computational simulation, and more. During his PhD, he focused on mathematical modeling and solutions of differential equations. Sir has received awards, delivered talks, and is actively involved in mathematical and scientific societies. Today, Sir will be talking on the topic stability of a within-host model of dengue virus transformation. Without any further ado, I would like to call upon Dr. P. Muthu onto the stage. Sir.
No, it's okay. It's okay.
Hello. Hello. So good afternoon to all of you. First of all, I thank the organizer for inviting me to give this lecture. I'm very happy to see the students of second year MSc and other participants. So I'm happy to present this work in this August audience. So good afternoon to all of you. Then, as he has mentioned my brief biodata, that basically I'm a fluid mechanics, biomechanics scholar. Recently, I mean, 1 or 2 years back, I was motivated to work on this problem, like this mathematical modeling of epidemics. In fact, I was motivated during the like this workshop kind of things in IIT Madras in 2019 December. It was a 6 days program on the mathematical epidemiology conducted by Professor C.H. Srinivasa Rao. So one of the lecturer was my guide, Professor Piyush Chandra. So then I was got motivation after attending the 6 days program on the mathematical epidemiology. So then I was decided to work on some problems on the epidemic models. Then I asked my Guruji. Then he mentioned that you can start with some modeling of dengue fever or kind of HIV infection, which Professor Prasant Kumar Srivastava worked. So you can talk to him like that. He was given advice to me. Then I have explored the idea of working this. Then luckily, one of my students also got interested working on this kind of problem. He doesn't want to work on the fluid mechanics problem. So it is the match of the guide and the student, because I was looking for some change, and that fellow also doesn't want to work in fluid mechanics. So the match was well. So then we started working on that. My student is Bikas Modak. Probably he's watching through YouTube channel probably. Okay. So therefore, it is because of him I also started working on this problem. So this is the background of working on this mathematical modeling of the epidemic things. Of course, in particular, because wanted to work on the in vivo model rather than the working with the populations. So therefore, so this is what the idea. So why I'm saying this information is that probably one of you or some of you probably get motivation to work on this kind of problems and see the applications of the mathematics. See, it's not that just I want to work on and get something. My mind was to learn something. What is there in this kind of things? Curiosity, simple curiosity, to learn something more of mathematical things and see the applications. So I hope all young minds in coming days, you may also explore the various aspects of the mathematics. Yeah, so this is the background. Okay. So my talk will be on this kind of things: introduction, mathematical formulation, analysis, local stability analysis, global stability analysis, numerical simulation, sensitivity analysis. I put a star just to tell you that we have, in fact, because worked on many of the aspects of the modeling, but here I just present simple results without going to much of the sensitivity analysis, optimal control, and many other aspects. Of course, some conclusion and acknowledgement. References. Introduction. Just as my title says that I would like to study how the dengue virus spreads in the human body. So I just request to play the video with a little more sound, okay? This video just tell you how biological things happening in your body when the dengue virus attacks. It's not just only dengue virus. Many of the diseases when we have, this is what the process in general. This is a particular process, which I am going to give the video as a particular process, how the dengue virus affects the body. Of course, it is available freely on the YouTube. If you put it there, life cycle of the dengue virus, you will get this video. But just for the brief of the for the audience, I'm just playing it here.
RNA virus. Its outer surface is covered with envelope proteins.
You may not follow
surrounding a lipid
all technical terms.
bilayer
Don't worry,
envelope.
because you just see the
Inside
process,
envelope
how
is a
the
capsid shell that contains
particles
the
move.
virus's
Virus
RNA
attacks the and how it reproduces. Just observe the
Immune
overall process.
cells are
Forget about
targeted
the
by the dengue
details.
virus. There are two cell surface receptor molecules important in dengue infection. The cognate receptor is involved in normal infection, and the FC receptor is involved in a phenomenon called antibody-dependent enhancement. The virus's envelope protein binds to the cognate receptor and triggers a cellular process called receptor-mediated endocytosis. The virus is internalized in a bubble-like structure called the endosome. When endosomes form, proton pumps lower the pH bacteria. The virus responds to the lowered pH by changing the conformation of the envelope proteins to form spike-like structures. The tips of the spikes are hydrophobic, which allows them to penetrate the endosome's membrane. They bend until the endosome's membrane and the virus's membrane fuse together and release the capsid into the cytoplasm. The capsid breaks apart and releases the viral RNA. The viral RNA travels to the rough endoplasmic reticulum. It is a positive sense strand and can be directly translated into proteins. The ends of the RNA form structures that bind to translation initiation proteins. The complex attaches to the ribosome to initiate translation. The whole viral genome is translated as a single long polyprotein chain. The capsid protein is on the cytoplasm side of the endoplasmic reticulum. The envelope protein and the pre-membrane protein are in the lumen side and are activated by the host's peptidase enzyme. In the cytoplasm, one of the viral proteins, a protease enzyme, activates all the other proteins in the polyprotein chain. These proteins aggregate to form the RNA replication complex. The viral RNA is synthesized in multiple steps. First, the ends of the viral RNA fold up and the RNA forms a circle. The RNA then attaches to the replication complex to start the first round of synthesis. Using the virus's positive sense RNA as the template, a negative sense copy is made. The pair of RNA strands forms a double helix. The RNA then becomes a circle again. This time, the negative sense strand acts as a template to make a positive sense strand. Many copies of the positive sense RNA are made by repeated cycles of RNA synthesis. Some of these strands are translated to make more viral proteins. Eventually, enough proteins are made to assemble new viruses. The envelope proteins aggregate in the lumen of the endoplasmic reticulum, and the capsid proteins aggregate on the cytoplasmic side. A viral RNA binds to the capsid protein and is packaged into a new virus particle as it buds off into the endoplasmic reticulum. The virus is still immature. Its pre-membrane proteins cover the tips of the envelope proteins to prevent premature fusion back into the cell. The virus buds off and travels through the Golgi apparatus and continues toward the cell surface. Before reaching the surface, the pre-membrane protein is processed and the virus becomes mature. New dengue viruses are released from the cell, ready to infect other cells.
I hope you might have observed a certain biological process happening in the body when the one dengue virus attacks us through the skin, whatever by the things. In fact, dengue virus is spread by the bite of the mosquitoes. I will tell the details here. I have already mentioned here. The whole process is of course, whatever I have seen in this video is nothing but the overall process. To model the overall process it's not easy. I mean, it's very difficult to model the overall process since the bite and then how the molecular DNA or RNA, all the process, then how it replicates, and this whole process, it takes almost, I think I've written here, that it takes around 7 to 12 days to bring the dengue fever. Infection, I mean, first attack, then bringing the fever. So this takes a overall process. What you've seen is, as you mentioned, that one of the at one point of time, that it takes some time so that it attacks and replicates, and it reproduces. So this all, you can see that the verbs translates, attacks. These are the verbs are used to translate to mathematical modeling. Please make a note that whenever you see the problem in a real life problem, you have to observe what are the verbs are used to describe the problem. The verbs will be the action in mathematical things. So if you see that like that, there are so many other things. Whatever you see in the video are given in a single picture, the whole process. So as people say, one picture is worth of 1,000 words, like that. This picture is the whatever the words are described by the video in simple terms. You can see here the one which is first virus. It is entered to the body and cell things processing, and the RNA all other processing. Then it attacks the what you call DNA. Then we miss a dengue virus protein translation, and these are the replications, and then assembly. Then they matured, and then the cover, you have seen the cover, and then when it come out, then it is released into the viron. So the whole process is a kind of few days. Say, as I mentioned, a few days it takes time to do these all operations in your human body. So this is the kind of idea, but as I mentioned that modeling this whole process, it will be very difficult. So the dengue fever is a mosquito-borne tropical disease caused by the dengue virus. Symptoms may include high fever, headache, vomiting, muscle and joint pains, and a characteristic skin rash. Sometimes in some cases, more severe dengue hemorrhage fever or dengue shock may occur, which can be fatal. Dengue is now endemic in 128 countries, and it strikes as many as 96 millions of people each year. Dengue virus is an RNA virus of the family Flaviviridae or genus Flavivirus. There are 4 types of serotype, dengue 1, 2, 3, and 4 of the dengue virus. The 5th type of the dengue virus is discovered in the 2013. Dengue virus is transmitted by the mosquitoes, as I mentioned already. In particular, this name, Aedes aegypti. That's what even in these recent days you might have heard in the newspaper. In particular, I think Bombay and in Tamil Nadu, certain areas, this fever is spreading because of the stagnant water. Because of this virus, this is mosquitoes. These mosquitoes breeding this kind of disease. In this regard, a few articles are discussed. Certain literature, I just present you for the information. Of course, whatever I have presented is very few. Actually, there are so many literature. Just for few, I brought it here to your notice. So this is one of the some paper. It's given you the dengue fever in a clinical review, and these are the details of the paper. This says that based on a severity of infection, we can separate patients into the groups: Group A, Group B, and Group C. I can see that the patient with no warning, because it may not give the warning immediately. That's what I told now. It takes some time to spread inside the body, the cells. And patients with the developing warning signs with the coexisting risk factors for serious infection. Then C is the serious one, the severe one. Paper 2, you can see the Diamond and et al. This talks about the molecular insight into the dengue virus pathogenesis and its application for disease control. Again, these are some of the observations. Antibody-dependent enhancement during secondary infection results increased viral burden and more severe symptoms. And some of the other details, I just leave it to you for your look. And this is another paper by Mukherjee et al. The title is that the mosquito immune system and the life of this dengue virus, what we know and what we don't know. So this is the pathogens, and these are the certain observations. And it says that the virus resides as a common cell in its vector, and the physiological effects, physiological effects due to viral propagation in mosquitoes are different from those in the human system. This says that when the dengue is with the mosquito, it may not affect the mosquitoes, but when it bites through the human, then it affects starting. The virus has a known detrimental effect on the mosquito itself, which remains infected for life. Though it may be having the viral things, but it may not be infected by the mosquito. Dengue is the fastest spreading mosquito-borne viral infections disease worldwide, with a remarkable morbidity and mortality. In India, the first epidemic of the clinical dengue-like illness was recorded in Chennai in 1780, and the first virologically proved epidemic of dengue fever occurred in Kolkata and eastern coast of the India in 63-64 time. Then the fourth serotype is detected in Pune, Maharashtra, after an absence of 30 years. In the recent means 2010. As I mentioned in the beginning, the Maharashtra and Tamil Nadu, there are some issues with respect to dengue fever. Of course, classification of mathematical epidemiology, one can study the dengue virus with respect to the in vivo studies or animal studies, and another group of studies, the in vitro studies or cellular studies, and the human population. That's what epidemiology. Though here we are focusing on this aspect. That is, we are focusing on the in vitro studies or the cellular studies. You can look at this literature, Noorani et al. It is the title is the mathematical model of dengue internal transmission process, published in the Journal of International Mathematical Society in the year 2007. So we, as we decided to work on the dengue fever, and in particular the internal transmission of the virus, then we got this paper, and this is what our starting point. We have started with this work. We have worked out the complete steps of this paper to get the better knowledge of what's happening in the mathematically. As any one of you know that to start the research, you start with a single paper, yes or no? You work out and see the learning the basic steps. So this is what our base. In fact, this is the first work of my student. So in this article, we will discuss about the internal transmission of the dengue virus in a human body. In order to derive the equations of the mathematical model, it is assumed that dengue viruses are virulent and no other microorganisms that attack the body. So that's what this idea. As you can see that Noorani et al. They present a mathematical model. We also look at the stability analysis. Of course, they have done a very basic analysis, but we have extended to many more ideas. So the objective of this study, the work is that we present a within-host model that describes the infection caused by the dengue inside the human body. It is similar to that of the model given by the Noorani, but the derivation of R0 differs. In fact, when we work on this problem, the R0 given by the Noorani and R0 we have calculated that it differs in certain terms, and then not only the terms but also while applying this R0 to study the effect of the various things on S or I and other things, we found that there are a lot of differences. Therefore, the derivation of R0 is not, of course, I am not saying that wrong, but it has certain issues with the Noorani paper. So we are trying to rectify that issues. So the R0 given by them gives a higher value, leading to the understanding of abnormal production of the newly infected cells, which is not the case actually when we look at that. So this is the okay. I have given a video. I have given a one-page figure. Now you look at that. What we are doing it actually here. Here we are taking it to simpler model, though the whole process is quite complicated. But we have taken only 3 compartments to start with. As I mentioned that the whole issue is here. Our basis for this paper. So therefore, we are working on according to this model with these 3 compartments. Here, the SIV are not human populations. The SIV are cell populations. So therefore, the S indicates the susceptible cells. I indicates the infected cells. And V indicates the virus load or the virus particles. So you can see that there's a constant production of the alpha, the constant production of the uninfected cells by this rate. And of course, this is the natural death rate of S. And this is the susceptible cell interact with the virus. As I mentioned in the what you called it, the picture, that virus and healthy cells, they interact and then they also reproduce, or some of them are dying. So therefore, all aspects of the how the virus is replicating inside the body are taken care by this simple 3 compartment. Of course, you can have various other aspects also by adding few more compartments as you wish from the biology. These are the interaction process. The I, you can see that the I compartment to V, this is the what you call the virus produced by the infected to the virus. So the infected cells become virus also, and this is the natural death of the viruses. So this is the overall transmission diagram of the dengue virus. You can see that the dynamics of the cell population is given by the following simple equations. That is ds by dt equal to alpha minus beta into SV minus delta ST, and di by dt equal to beta into S into V minus sigma I, and dV by dt is equal to Ki minus gamma 1 V minus gamma 2 V minus beta. This indicates that the interaction of the unhealthy cells, healthy cells and the virus. So this is a taking care of the appropriate modeling based on the assumptions which I mentioned earlier. So here S is the susceptible cells, I is the infected cells, B is the free virus particles, alpha and delta are the production rate of the uninfected cells and death rate of the uninfected cells respectively. Beta is the infection rate of the uninfected cells due to viral cells. K is the rate of production of the new free virus cells, and gamma 1 plus gamma 2 is the rate of removal of virus cells, where gamma 1 is the natural death rate and gamma 2 is the death rate of the virus due to T cells. T cells is like a kind of antibody cells. Sigma is the death rate of the infected cells. So based on this understanding, this is the model 1. And we have model 1. What do you want to do with the model 1? How the the SIV are behaving with respect to time as well as with respect to these parameters. I have given the parameters, almost say 8 to 7 parameters are there. So how this varying, how these dynamics of the S and IV with respect to this thing we are going to look at. First, we want to see that the feasible region of this biological interest of this model is that where this omega, where SIV are greater than or equal to 0. It means that we are in the three-dimensional model. Yeah, the feasible region is positively invariant of the system 1, since the vector field on the boundary of omega does not point the exterior of the omega. We have calculated the equilibrium states. E1 is the virus-free equilibrium state. This E1 given by alpha by delta 0, 0. The equilibrium state of abundance of susceptible cells, infected cells and free virus is given by E2, that is S star, I star, V star, and these are given by this one. I think many of you know the procedures how to get these all things. The basic reproduction number R0 is nothing but the meaning already has given that how one infected cell will produce a new infected cells. That's depend upon the R0, that reproduction ratio. So to get the value of R0, we use the next generation matrix method. The R0 is obtained from the spectral radius of the following next generation matrix, the P, where gamma is nothing but this one. The matrix P is obtained from the Jacobian of the equations di by dt and dv by dt of the given model. It is evaluated at the virus-free equilibrium point E1. The basic reproduction number of for this model is this one, and you can see that the meaning of it is given here. Biological background of the R0. One infected susceptible cell produces K by epsilon virus particles during its infected period 1 by sigma at a constant rate K. One virus particle infects this many susceptible cells during its active period, where this is the rate at which the susceptible cells are infected by the free virus particle at its equilibrium value alpha by delta. This is the virus-free equilibrium state. The geometric mean of these two terms gives the basic production number, a basic reproduction predicted number R0. To determine the local stability of the equilibrium state E1, the Jacobian of the equation 1 is evaluated at E1, which is given by this one. And to prove that this is locally stable, so we have this theorem. The virus-free equilibrium E1 is locally asymptotically stable if R0 strictly less than 1 and is unstable if R0 greater than or equal to 1. And for this, the eigenvalues of the Jacobian matrix D1 are calculated, and they are nothing but minus delta and the roots of the following quadratic polynomial. So this is the polynomial. You have to find out the roots of the lambda, and these are the parameter values, sigma, all other things are parameter values. The roots of the quadratic polynomial was given by this one, and after looking at the critical analysis, we got this outcome. The local stability analysis of E1, that is, when R0 less than 1, the roots of the polynomial have a negative real parts, and when R0 greater than or equal to 1, one of the root has a non-negative real part, the other one has a negative real part. Hence, E1 is locally asymptotic whenever R0 strictly less than 1. This the end of this concept of the local stability of E1. Next, the equilibrium of E2, I mean the local stability. The equilibrium E2 exists if R naught greater than 1 and it is locally assembled stable if and only if it satisfies the following condition, this condition, the case number 4, where R naught bar is nothing but this one. How you do that? We do the same thing, similar process. Substitute the expression of the basic reproduction number R naught in this E2 and we apply these following things, S star, I star, V star, in terms of R naught bar, where R naught bar is given by like this. And here the S star, I star, V star are positive if R naught greater than 1 or R naught bar greater than or equal to 0. To determine the local stability of the equilibrium state E2, the Jacobian of the equation 1 is evaluated at E2, which is given by this one, where gamma is nothing but gamma 1 plus gamma 2. The values of the S star, I star, V star are all given by the equation number 5. Now the local stability answer for E2. The eigenvalues of dE2 are the roots of the following cubic polynomial, that is S cube plus A1 S square plus A2S plus A3. The A1, A2, A3 are to be given by this way, the following expression, in terms of the parameters of the model. We apply Routh-Horvitz stability criteria to prove the local stability for the E2. Let us consider nth order homogeneous system of equation. Just I just give you the background of the Routh-Horvitz stability criterion. So if you have this system, I think then I can skip the Routh-Horvitz stability criteria. If you have this system, you can have a stability analysis based on the roots of this nth order polynomial, where if the roots are, if you look at this polynomial, the necessary condition for the stability is satisfied if all the coefficients of this are positive. Hence, we assume that all the coefficients should be of this one. So first we take it as A naught as 0, the first coefficient. We write the Horvitz matrix as follows. Based on this matrix formation, this is the condition, and there is a theorem by the Routh-Horvitz. This is the theorem given by Routh-Horvitz. If you satisfy this condition, then you are proving the local stability for the E2. And in our case, it works out. And based on that analysis, we conclude that the one that is the local stability for the E1 and E2 are proved through respective methods. To prove the global stability of the points, we have used the Leibniz's direct method, which is given by the following way. To establish the global stability of the virus-free equilibrium E1, we use the Leibniz's direct method. We propose the following theorem to examine the global stability of the virus-free equilibrium E1, and this is given by this theorem. As theorem says that R naught less than or equal to 1. If this is the true, then the virus-free equilibrium E1 of the system 1 is globally assumed stable on the omega. Of course, we have taken this what we call Leibniz function, and we have proved that the derivative with respect to t, it's a negative. So that we see that L1 dash is negative if R naught is less than or equal to 1. And we note that L1 dash equal to 0 if and only if S is equal to S star, I equals to 0, V equal to 0. So therefore, by Lassalle's extension to the Leibniz method, we prove that virus-free equilibrium point is globally assumed stable on omega. Similarly, for the E2, a similar procedure, we have taken a different Leibniz function, and the similar analysis repeated, and we got this analysis. Therefore, endemic equilibrium state E2 also globally assumed stable on omega. And these are certain graphs with respect to for some particular set of parameters to verify that yes, we are giving a negative values. So therefore, x-axis time, y-axis L1 dash. Similarly, t versus L2 dash. Of course, we have taken the t versus s also for a set of some parameters of interest. Just to show you that some numerical simulation here. So the numerical solution of the SIV with respect to different beta are also just given to have how the behavior of the system with respect to beta we are just given. As I mentioned that we have not given the sensitivities of the parameters. Of course, we have done. I have the paper. If anybody want to see it, I can show the sensitivity analysis of the parameters also. So this is we have written in a paper, and in fact we have modified certain aspects of this work to include some more effects, and we are in the process of sending to your paper. So this is called as stability of a Wittig-Gross model of Dingwest transmission. These are some papers. as I mentioned, Nurani is the things. I would like to thanks my institute who has given my leave also, permission to go and come back here and present this work. And in fact, I request to all to look at the NID Warangal website, look at the mathematics department. We have 4 different kind of programs: MSc 2 years applied mathematics, MSc 2 years of maths and scientific computing, 5 years integrated mathematics. Now we are in this academic year we are offering 4 years BTech program, maths and computing, and as well as BSc B.Ed also. Government of India, Minister of Education asked NIT Warangal to start this work of BSc B.Ed, which is a 4 years course. So therefore, these all 5 courses under the mathematics department. I request you to have a look at the NIT Warangal website and have a more information. Not only on the institute, look at my website. If anybody interested, I am ready to interact with the mathematical biology also. As I mentioned that, Professor Piyush is my inspiration. In fact, when I was started this work, we were having certain doubts. We are unable to move forward certain technical details. So I called Professor Mini, Professor Prasan, over a phone, over WhatsApp, over Google Meet. So they spared some time for us. Okay? So it's really great that they helped us. And this is because he is doing now with me PhD. In fact, you will submit soon the thesis. Thank you.
Thank you, sir, for traveling a lot from Warangal. There are lots of rains in Telangana if you are following the news.
I forgot to tell one.
Yes, yes.
We have met a few weeks back only. Okay, na, in Jeju Island, South Korea. Okay, last week of the June. Okay, I never know him earlier also. Of course, I heard the names, but we never met. But after attending the 5 days conference, and all the models, most of the problems discussed in the Korea conference are nothing but this: whatever you do is in 3 days. Okay, na? And of course, I should tell one very important thing is that China, Japan, and Korea, these 3 people are like anything doing like anything research work. I can't quantify. If you want to ask Ritik, okay, he will tell how much work they are doing it in a qualitative way. Okay, na? Nowhere we are. After looking at the Indian scenario, after observing here and then other conferences like this, we are nowhere. I mean, probably I am now only started. Probably, maybe after some time, maybe maybe you can give some comments on these aspects.
So we are also progressing.
Yeah, we are also trying to the best.
That's the idea.
So this is called as a China, Japan, India, Korea conference. 4 countries, a colloquium, like what he wants to create here, a group here. That's the international level. Okay, this is the national conference. So therefore, thanks to Vamsi, after interacting me, he immediately called me this conference. Thanks for the invitation.
Thank you, sir. Just at such a short notice, you have traveled in spite of heavy rains. So any quick question from the participants? Any from virtual? Do we have any? So let's thank sir.
I have email address. You can write to me.
Yeah, one question, please. We can quickly take. Yeah, what's the question? Yes, just quickly unmute them. Can we hear them? Okay, leave it. We'll move on to that. Adam, yeah. Can you, I don't know their name, so can you kindly put in the chat box? We'll pass it on to Muthu sir. Okay, let's give a round of applause for Muthu sir. For though sir is a senior, but is almost like a okay brother or a friend. So we have kind of interacted. Ritvik also knows he is very close to sir. They almost stayed next to each other in the adjacent rooms in the same hotel, right? Okay.
Thanks for everyone for just listening. After lunch, in fact. Thank you. Actually, don't mind. The video was intentional. Not that you should understand. Why you put a video there so that you wake up after lunch.
Actually, next speaker sir has already given an introduction.
Thank you, Professor Muthu, for the insightful presentation. Now, let me introduce you to the next speaker of the session, Dr. Mini Ghosh. Professor Ghosh is a member at the School of Advanced Sciences, Vellore Institute of Technology in Chennai, India. Professor Ghosh earned her doctorate in mathematics from IIT Kanpur in 2002. As a postdoctoral research fellow, she conducted research at the Department of Mathematics, University of Trento in Italy from 2002 to 4, and the Institute of Information and Mathematical Sciences in Massey University, Auckland, New Zealand. Ma'am is actively engaged in various research problems with specialization in mathematical modeling of epidemiological and ecological systems, and will be delivering her talk on the topic Mathematical Modeling of COVID-19. Please join me in welcoming Professor Mini Ghosh.
Thank you very much. Thank you. Am I audible? Hello? Yes, am I audible?
Yes, madam, you are audible, madam.
Yeah.
Can you request to
Good
kindly make the
afternoon, yeah.
screen full in presentation
Okay,
mode,
okay,
madam?
yes.
It
Yeah,
will be
yeah, I
better
will.
for us. Full screen mode. Yeah, it's better, madam. Thank you.
Yeah. So good afternoon to one and all present here. So first I would like to thank the organizer for giving me opportunity to present our work in virtual mode, especially Dr. Bamsi. And so I'm going to talk on mathematical modeling of COVID-19. And this topic is not new. In fact, all of us know about COVID-19, and also there are thousands of models. Okay, so most of you are familiar with the modeling COVID-19, but here I will just discuss about some of our work which we did in our group with the help of our PhD students. So this talk is outlined like this. First is introduction. So here I will say a little bit about epidemiological modeling. Then I will come to basic reproduction number, which is very, very important in disease modeling. And then because in one of our model we got backward verification, so I will just discuss in brief what is backward verification. And we have included treatment function in our model, so I will discuss little bit about different types of treatment functions. Then facts about COVID-19, I may omit this because all of us know about it. Then I will take one model on COVID-19 by incorporating treatment function. If time permits, then I will go to another model, that is the modeling COVID-19 with the impact of awareness. And then finally, I will say a little bit about recent trends in modeling COVID-19 and issues and challenges to model COVID-19. Okay, so first thing is, why do we need epidemiological modeling? Mathematical modeling of infectious disease helps in understanding the disease, in analyzing different control strategy in the same population or different population. It is used to identify trends and making general forecast about the spread of disease. It helps in evaluating the potential impact of an epidemic. In fact, repeatable experiments and actual data are not available for diseases, so in fact infectious diseases. So mathematical modeling is very useful tools, and we can play with the parameters using simulations, and we can predict better, means we can give better prediction for the transmission of the disease. So there are several factors, several parameters which influence the dynamics of infectious diseases. First is susceptible population size. In fact, first we should know what is susceptible population. So if we are talking about the childhood diseases, then only children are susceptible. If we are talking about HIV, then obviously only adult populations are susceptible. But if we are talking about influenza, TB, and COVID, then we all are susceptible. So susceptible population should be there, then only we can talk about the transmission of the disease. And size of susceptible population, that is also very important, because it should have some threshold, otherwise disease will not persist in the population. Then mode of transmission, that is also important, because how it is getting transmitted, whether it is direct transmission or indirect transmission. Homogeneity of population means whether population is homogeneously mixed or heterogeneous. Infection transmissibility, how fast it is getting transmitted, that is also important. Then we have immunity level of individuals. So different individuals will have different immunity level, and depending upon they will show the symptoms or they will not show the symptoms, and there will be delay in the onset of the symptoms. Movement of individuals, when individuals moves from one place to another place, disease gets transmitted. Then infection incubation times, that the time when the person is exposed to the infection and when the symptom appears. Okay. This time is also very important. There are several other factors like environmental, demographic factors, geographic and economic factors. Some environment is very conducive to the growth of mosquito population. So in the case of dengue and malaria, etc., environment also plays important role. And economic factors, you can see that if someone is not able to afford the treatment, then they will delay in consulting the doctor, and that delay can cause the spread of disease in the third level. Then infectious period, which is very, very important. So different diseases will have different infectious period. If infectious period is long, then that means that person is capable of transmitting the disease for longer period, and that can lead to more transmission of that disease. Then come to the basic reproduction number. As Dr. Muthu already said, he was discussing about in-host dynamics, but we are talking about the population dynamics, but it is defined in the same way. So this is nothing but the average number of secondary cases generated by a single case in his or her whole infectious period. That means when you put one infected in a fully susceptible population, how many new infections are getting generated due to that primary infective, that number is called the basic reproduction number. And in fact, in classical epidemic models, you will see that if this number is greater than 1, then only disease persists in the population. And if this number is less than 1, then disease dies out. That means that if one infective is not capable of transmitting the disease to one susceptible population, then obviously disease has to die out. So for a simple model direct person-to-person transmission, this R0 is given by rate of transmission times infectious period times the population size. That is the product of rate of transmission, infectious period and population size. So when we find our basic reproduction number, then if this number is say greater than 1, then our control policy should aim to make it less than 1. If it is say 1.5, then control policy needs to eliminate a fraction of transmission, that is 33% for R not equal to 1.5, so that it will be less. If R not is 2, then control policy should aim to reduce the transmission by 50%, so that R not can be made less than 1. How to achieve this? There are several methods. So first is reducing contact, that is quarantine, increasing social distances, etc. Reducing susceptibility through vaccination, reducing infectiousness through treatment. Okay? So we all know that if we do all these things, obviously we can reduce or we control the transmission of infectious disease. But the question is that the issues are that who are being targeted, how much effort is needed, and how fast do we need to act. If we want to know all these things, then we must formulate a good model, and then we can do better prediction. We can tell something about the transmission of that disease in later span of time. And then why do we care so much about R0? R0 is the threshold parameter which determines whether disease will persist in the population or not. R0 determines the initial rate of increase of an epidemic. It also determines the control effort required to achieve eradication. When we formulate the model, our expression for R naught will involve the model parameters, and those parameters will be related to control effort, rate of transmission, recovery rate, all those. So we will come to know that which parameter we need to increase or decrease so that we can make our R naught small. In classical epidemic model, what happens here is x-axis we have say R naught, and y-axis we have equilibrium level of infected population. This is just a rough sketch just to demonstrate what is forward bifurcation and what is backward bifurcation. So in first graph, you can see that when R naught is less than 1, we have disease-free equilibrium point here, and when R naught is greater than 1, it is moving like this. We have endemic equilibrium point, unique endemic equilibrium point. In the second graph, you can see that we have disease-free equilibrium point on x-axis when R naught is lying with 0 to 1, and we have this backward movement and that is called backward bifurcation. So this implies that when R naught is less than 1, in first graph you can see that only disease-free equilibrium is there. But when R naught is less than 1, in second graph you have disease-free equilibrium point as well as you have endemic equilibrium point. If you draw one vertical line here, you can see that you have two disease equilibrium points and one disease-free equilibrium point and two endemic. So in this case, what happens that it depends upon system may tend to disease-free equilibrium point or system may tend to endemic equilibrium point depending upon the initial population size. That means if you are in the neighborhood of endemic equilibrium point, then system may tend to endemic equilibrium point, and if you are in the neighborhood of disease-free equilibrium point, system may tend to disease-free equilibrium point. That means you can't guarantee that disease-free equilibrium will be locally stable or endemic equilibrium point will be locally stable, because you have more than one equilibrium points depending upon the starting initial start. It will depend which equilibrium point will be stable or system will tend to which equilibrium. Okay, so in this case you can see, okay, I will come to my model, then I will say a little bit more about this. Okay. So now come to different types of treatment function. So here you have linear treatment function, which is say T of I equal to alpha I. That means that rate this rate of treatment is linear. That means it is proportional to the number of infected. Second one, it is introduced by Wendy Wang in 2006, and this is space treatment function. So if number of infected is less than some threshold, then it is linear. Otherwise, it is constant. And that constant is nothing but the maximum capacity, that is R I naught here. So treatment rate is proportional to the number of infected when the capacity of treatment is not reached, and otherwise it takes the maximal capacity. So we have incorporated this in a simple SIS and SIR model in 2009 or 10 with my Chinese collaborators. But then we thought to incorporate this COVID model, and that we did. And we showed that there exists backward bifurcation. And there is another type of treatment function, that is a saturated treatment function. This was introduced by Hang and Liu in 2008. And it says that for when number of infected is small, T of I, that treatment function that is proportional to Ri. When I is large, then T of I is equivalent to R upon alpha, when I is tending to infinity. So this characterizes the saturation phenomenon of the treatment, and this function is continuous and differentiable. And obviously, when alpha is 0, this treatment reduces to the linear one. Okay, and this is Holland type 3 treatment function, which was introduced by Dr. Dubey for the disease modeling. And here it is, when I is very small, that rate of treatment that will be very, very fast because of this second power I square here. Okay, and but when it increases I increases, then finally it grows slowly and finally settles down to maximum saturated level. So in this case, beta upon gamma. And this condition pertains to the known disease which has re-emerged and has available treatment modality. So we can talk about this for COVID-19 also. In fact, I have seen one model by Dr. Bansi. I will just display that on. Okay, so these are some facts about COVID-19. I think I can skip this slide because we are already running late and all of us are aware of all these facts. Okay, so now we have crossed more than 44 million in the number of cases of COVID-19 in India. Okay. So I will come directly to our model, and this is a joint work with PhD students Akhil and Shraddha. Now they are doing postdoc abroad. So I must thank both of them for helping me to work on this model. Okay. So we are here talking about method of modeling of COVID-19 with limited public health resources. In the sense that we are going to include that treatment function, stage treatment function, which was first introduced by ND1. So we have a compartment model. We have considered three most affected countries, USA, India, and Brazil, and we have fitted our model with data from these countries. First, I will discuss the compartment model. So here we have different compartments. We have susceptible, exposed, infected compartment. We have asymptomatically infected and symptomatically infected compartment. This is Q that denotes QI, that is quarantined people. It could be home quarantine or institutional quarantine. Then this is the individuals who are admitted in hospital, and this is the recovered compartment. We have also introduced that non-pharmaceutical intervention that is related to use of face mask, maintaining social distance, etc. So here this is a decay factor. You can say that that is reducing the number of susceptibles who are exposed to the infected. So we have 1 minus sigma MPM. This product term is coming because of efficacy of those measures. That fraction is sigma m, so that is why, in fact, this product as a whole is less than 1. Okay, so this reduces the transmission. Okay, so we have assumed different rate of transmission due to asymptomatically infected individuals and symptomatically infected individuals, and which are given by here beta 1 and beta 2. Okay. So from when susceptible interacts with infectives, they move to exposed compartment. From exposed compartment, some fraction will move to the asymptomatically infected compartment, and some fraction will move to symptomatically infected compartment. And from asymptomatically infected compartment, if they show the symptoms, they may move to the symptomatically infected compartment, or they may go to a quarantine group. And from there also, if they show some symptoms, they may move to this compartment, symptomatically infected compartment. And from symptomatically infected compartment, they go to the hospitals if they take the treatment. So this is treatment function we have included only for symptomatically infected individuals, because those who are asymptomatically infected, they don't require any treatment. Okay? Their symptoms are very mild, or they may ignore this. So here you have H compartment. From H compartment, they may recover and join a recovered compartment. From this home quarantine individuals or institutional quarantine individuals, they can also recover and they can join this R compartment. So we have natural death rate mu in all the compartments, and we have additional death rates in only I, S and H compartment, those who are symptomatically infected and those who are hospitalized. So mu S and mu H, and these are the rate at which they move from one compartment to another, and corresponding system of differential equation we get like this. And here we have T of IS, the treatment function, which I have included here. In fact, we have included here, and because of that, we have when T of IS is some alpha IS, we have say P of IS is alpha S IS when IS is less than some threshold I naught, and this is equal to alpha S I naught or K when it is greater than I naught. So this thing we have included in our model, and Then we analyzed our model. We get say disease-free equilibrium point like this. This is the base reproduction number by using this generation matrix method. First corresponds to the transmission due to the asymptomatically infected individuals. Second term corresponds to the transmission due to symptomatically infected compartment individuals. So beta 1, and here we have beta 2. So this is our base reproduction number. Then come to the existence of endemic equilibrium point, where we have when IS is less than or equal to I naught, we have this alpha as IS, and when it is greater than I naught, we put here as K, some constant. Okay, so we have this kind of system, and when we have IS less than I naught, for R naught less than 1, we have only DFE, and R naught greater than 1, we have only endemic equilibrium point. Okay? So I will not go in much detail. When IS is greater than I naught, then we have, say, all other variables we can write in terms of E star, which is corresponding to exposed individuals, and E star is the positive root of the following quadratic equation. So we have this quadratic equation. As this is a quadratic equation, it has two roots. So we have different conditions. Depending upon the conditions, we can get one positive real root, or we can get two positive real roots. So when C2 is less than this, we have one positive root. When C2 is greater than this, we have two positive real root, and corresponding to that, we have two positive endemic equilibrium point. Okay. So this suggests that there is a probability of backward bifurcation, and this is exhibited through this plot. So for this set of parameters, we just found that we are getting backward bifurcation like this. We our x-axis is your reproduction number. In fact, we have considered beta 2 as the critical parameter, and corresponding to beta 2, when we change our beta 2, our R naught is getting changed. So x-axis we have plotted R naught, and y-axis we have considered exposed population. One can consider different infected population also. So we just considered exposed, because infected we have two IS and IA. So we can merge both, we can add, but we just put exposed up, and we saw that we have this kind of backward bifurcation. So here you can see that if you reduce R naught below 0.39, then only we have only disease-free equilibrium point. Okay, between 0.39 to 1, we have disease-free equilibrium point. We have this endemic equilibrium point. We have this purple, this kind of here also we have endemic equilibrium point. So depending upon the initial start, system may tend to E2, or system may tend to E0. E0 corresponds to DFE, disease-free, and E2 corresponds to endemic equilibrium point. In between, you have unstable endemic equilibrium point, that branch is there. Okay. Also, reducing R0 below 1 is not enough to eradicate the disease from the population. Okay. So, this is the thing. In general, we believe that, okay, if R0 we make less than 1, that is enough, but that is not enough. It depends upon the situation. Okay. So this is the same backward bifurcation graph, but here we have done this graph bifurcation diagram with respect to the I0, that is the capacity of treatment. When we increasing the capacity of treatment, we have little bit mild reduction in the number of exposed individuals, and then finally increasing it further doesn't affect much. We don't have, if you have enough capacity, then there may not be the exposed individuals. Okay, all are getting treatment, and we may get only here. So we did stability analysis for different equilibria, for disease-free equilibrium point, for endemic equilibrium point, and all these are stable under some restriction on parameters. So more details one can see on our published paper. They can refer. Okay, so we consider the data, because this is our initial work. So we had considered data of this USA, Brazil, and India for first wave only. From this period to this period, and data we had imported from this website, and we used our software, and we fitted our things, and then we estimated beta 1, beta 2, and alpha A, and value of R0 you can see here that for USA it is more, India is at second number, and Brazil it is least. Okay? So all other parameters we considered like this, and we changed this lambda, and we saw the impact of these parameters. Okay? So it is not the fixed thing. That is the rate of recruitment in the population, which one can change. Okay, so this is the model fitting, and this is the sensitivity analysis. I think x-axis is not visible properly. I will discuss in the. So you have a bar diagram, say forward and backward indices. You can see that in the positive side and negative side. So it just signifies that with the change in the parameters, how R naught is getting changed. So if you have this high, this bar, then you can say that, okay, this parameter is more impacting the R naught. Okay. So this is the same thing. This is the contour plot of R naught with respect to alpha S and alpha A. Alpha S, alpha A are associated with the rate of treatment, and say you can see here that this dark green, that means your it is R naught is less here. As you are moving to this lighter side, R naught is increasing. Okay, so if you increase alpha S and alpha A, your R naught is getting decreased. But here you can see that that decrease is not making R naught less than 1. Okay, so when we considered those data and when we estimated our alpha A, etc., so we found that it is not possible that disease will be eradicated from the population. So these are some the results which I already discussed, but if you want to see, just see that we have estimated beta 2, and it is very high for USA, and it is least for Brazil, and we are in between. Okay, so if R naught is least, that means it is that progression of the disease is little slower in Brazil. R naught magnitude increases with increase in the parameters values lambda, beta 1, beta 2, and A. And this happens because these parameters have positive indices with R naught. In a similar way, the parameter possessing a negative indices with R naught are sigma m, rho m, alpha A, mu, and mu s. So if you negative indices, that means if you increase that, and then there will be a decline in the R naught value. So this result suggests that increasing the value of alpha A and alpha S will decrease the value of R naught. Okay, so it suggests that better treatment for every symptomatic infected and institute and home quarantine policy should be implemented very carefully. Then only we can reduce the transmission of this disease. Okay, then we got backward bifurcation, that is stated here, that reducing R naught below 1 is not enough to obtain the stability of unique infection-free equilibrium or disease-free equilibrium point. So this was our result, and this paper I saw, so I just thought to include it because the organizer is from this institute. So here this is for stage-structured population, susceptible, infected, and recovered, who are young, susceptible, infected, and recovered, who are elderly individuals. And here we can see that for young people, only linear rate of treatment is included, or rate of recovery. Individuals are moving from I compartment to R compartment at this rate. So this is linear here. For elderly people, this Collin type 3 treatment function is included because they become more sick and their condition becomes more severe and they need hospitalization. Okay, so that is assumed here, and that is why only in this group of individuals this kind of treatment function is included. So I will not go in the details of this. I will just discuss in 5 minutes. I will just discuss about the models of COVID with impact of awareness. So what do you mean by awareness? Awareness about having infected with the disease, awareness about mode of transmission of disease, and awareness about current statistics. All these things impacts differently to the individual's mind. Okay? if you are having that disease or you are aware of that, then you will behave differently. If you are not having the disease but you know how this disease is being transmitted, then you will take the precautionary measure. And then if you see that, okay, daily news is coming that, okay, so many people are dying and there is no recovery or very slow recovery, etc., then you will get scared. Okay? So all these things one can incorporate in the model differently. Okay? So I will just say a little bit about the model only. I will not go in depth. So this is one more such model where they have assumed that awareness thing, but the thing is awareness, because of awareness, your if they are taking precautionary measure, that means use of face masks and social distancing, etc., then rate of transmission is being reduced. Okay, so this we only incorporated in our previous model. So this is the thing. This is by Sudhanshu Biswas and okay. So other things are more or less similar, but you have this reduction. Then I will say, okay, here they have assumed that disease is transmitted also by Q and H compartment, those who are quarantined and those who are hospitalized. But they have included this modification parameter depending upon Q1 and Q2. If Q1 and Q2 are 1, then there is no transmission because of Q and H. Otherwise, in hospital setting also, sometimes disease gets transmitted to the hospital staffs, nurse and doctor. Okay, so that we can incorporate like this. Okay, then we receive the second model where they have incorporated the impact of media. And this media compartment is included separately here. And susceptible individuals who are aware of the transmission of disease through media, they will interact with media, and that individuals will move to this action compartment. And these individuals in XM compartment will not become infected. They will not come to contact with the infected. So we have one separate group of individuals who are aware of the transmission of this disease. So this is in one way of incorporating the awareness thing. And then this is another model. Because of media awareness, this transmission is getting reduced. But this reduction is not a constant. In our model or in the first model which I said about the awareness, there we reduced it by some fraction. Okay. So there was some constant. But here reduction depends upon the intensity of the media. Okay. So this M, if it is large, things will be different. If M is small, things will reduction will be different. So like that, this is again saturated type things. So here they have made it a variable. Okay, depending upon M, this reduction is there. So this is one such model, and I will just discuss the model. I will not go in the detail. Okay, so this is our model here. So we have here susceptible, exposed, asymptomatically infected compartment, symptomatically infected compartment, and recovered, quarantine, and hospitalized. We have all these compartments. But here, this is the due to impact of media or impact of awareness. What happens that those who are symptomatically infected individuals, okay, so they may isolate themselves, or their neighborhood may come to know about that person is infected. So in this way, transmission due to those individuals will be saturated, will not be more. Okay. So that beta IAS, that is the first term, which is transmission due to asymptomatically infected individuals. That is of simple mass action incidence type. But here we have again used saturated type incidence, which is the reduction or decay in the transmission due to the awareness of having the disease or awareness of this transmission of the disease, and because they are detected individuals, they have symptoms and they may isolate themselves. So this is not the same like the product of ID and S. It is this saturation term is there. So this only we have considered here differently, and we consider the data of Nepal and India, and we compared, and we got this is the model. I will not go in detail. And we considered both first wave and second wave, and we saw that in second wave, R naught was little higher than first wave, and also in India, R naught was higher compared to the R naught in Nepal. Okay. And this is the curve fitting. This, I think, this was done in MATLAB. And this is the sensitivity analysis with respect to R naught. So different parameters will have different impact. So this is showing positive indices. Here you have negative indices. But the thing is, you have different parameters here. Maybe it will not be so visible. But the thing is that some parameters which had positive indices, that means increase in that will lead to increase in R naught. Negative indices, that means increase in that will lead to decrease in R naught. And if it is more, that means increase in that will lead to more decrease in R naught. Okay. But some of the parameters one cannot change, that is natural death rate and other things. But that rate of treatment, obviously one can change. Rate at which they move to, say, hospital, that is one that rate of treatment you can say, that is very, very important. A rate of recruitment, that is if you are consider the migration, etc., then in that way you can stop that thing and you can decrease the rate of recruitment in the population, that is lambda. That is also having more impact. So this lambda, this big one, that is lambda. This beta, transmission, rate of transmission is also having more impact. If rate of transmission is more, then R naught will be more. Okay, so these things are there. And we did some prediction till June 30, 2021, using second wave data for both the countries. And we found a decline trend for both the countries for the next 30 to 40 days. And we obtained higher effective contact rate for India in comparison with Nepal, that I already discussed. So these are the impact of different parameters, rate of hospitalization, rate at which they are moving to quarantine center, and that have a positive impact in the reduction of the disease. And then intervention related to medical facilities, quarantine centers, rapid testing. All these things means we have incorporated through the model parameters, and if we increase that, obviously that decreases the number of infections. Okay. We have more number of graphs in the published paper. I could not include all here. One can refer that. So this is the recent trends in modeling COVID-19. Recently I have seen some papers where virus population they have considered separately. In the initial page, we saw the simple SIR model. Then we found some complicated model, symptomatically, asymptomatic compartment, etc. Now they have considered virus population also separately, assuming that virus can sustain in the environment for some duration. So assuming that this virus population or individuals, some researchers, they considered it a separate compartment. Okay? And then co-infection model, we have also worked on modeling COVID-19 with TB and diabetes. It is not yet submitted. We are working on that. And there are different, with HIV also, people are doing co-infection of COVID and HIV. Modeling COVID-19 with incorporating vaccination, that also people are doing. Modeling COVID-19 with immunity. And discrete time compartment model based on fractional differential equation. And recently I saw some papers where reaction diffusion equations also they are including for COVID-19 model. Assuming that a special aspect of that means individuals, they move from one place to another place. And that also they are discussing global and local stability of that kind of model. And these are some challenges. First few can say that now it is not applicable because now COVID-19 that transmission is subside, not there, it is subsiding. So these things are not so important now, but mutation risk is still there. Fast mutation of virus is posing new threat, and vaccine may not be protection for new strain of this disease, so that may be possible. Also, because of that, we may face a problem in future, so we have to be very, very careful. And these are some of our published work on COVID-19. One may refer and one may explore through Scopus data. We have published more paper collaboration with Dr. Prasant. And obviously Akhil and Shraddha, they are my students. Now they are in abroad. They are also still collaborating with us. So I had got a project on COVID-19 from DST. So I must thank DST, Government of India, for supporting me, and thank you all for your patience.
Thank you, madam.
Thanks.
Am I audible, madam? Yes. Thank you, madam,
Yes, yes.
for taking time out
Yeah.
and giving a virtual lecture. Any quick question, because we are running little late? Yeah.
Yes.
Madam Jayaprakash, Dr. Jayaprakash Tripathi sir will ask a question.
Good afternoon, ma'am.
Yeah, good afternoon, sir.
So you have delivered very nice talk and different things you have discussed. Actually, have you also seen that how much your estimate is good, like a confidence interval have you obtained in the first model that you discussed?
That my student actually Shraddha did that part. In fact, in the first model, I think it was not so good. Second model, when we discussed Nepal and India, that model we had better things. Okay. That
Okay.
for second model we had better estimate.
Yes, yes. And actually, just I want to understand that if suppose there is some epidemic model and we have at the same time we have two endemic equilibrium, both are stable. So how we physically explain that physically, or what is it means, how we explain that in the terms of disease or in the terms of physical sense?
So have you obtained this kind of cases? Because in general what happens that if we have more than one endemic equilibrium points, then what happens that they will be stable under certain conditions. So at a time, both will not be stable, locally stable. When you have more than one equilibrium point, endemic equilibrium point, so you can talk about local stability of those equilibrium points.
Yeah.
They will be individually, they will be stable on different conditions. Okay, so one will be stable, then another will become unstable. I have not seen any model, disease model, where we have two equilibrium points and both are simultaneously stable. Okay, because in the case of backward bifurcation also, we have two endemic equilibrium points for R naught less than 1, but only one we found that is locally stable, another is locally unstable. Okay. So what you are saying, if you have obtained two for R naught greater than 1 or R naught less than 1?
So actually, I have studied some paper on the tuberculosis. So there is situation that two endemic equilibrium are stable at the same time.
Maybe it depends on, maybe madam, if you excuse me, I'll just add, it depends on where you start the initial conditions, sir. It's called as domain of attraction.
Exactly.
So which domain do
That
you
is
start,
fine.
that is where you get attracted.
Yeah, that is the local stability. But that what actually physically it means, why it is coming in the disease term, how
It
we
all
can explain
depends
that.
on where I start. Where I start, accordingly I get attracted to that point. So maybe just one argument. So due to
What sir is telling, Bamsi sir is telling that you can have the two locally stable endemic equilibrium point. But I have not seen that. Locally stable, that means if you start from the neighborhood of that particular equilibrium point, system will tend to that equilibrium point. So he is telling that there is a possibility, or you are also saying that you have seen some paper where two or more endemic equilibrium points are locally stable. You can't talk about global stability, but local stability. So depending upon the initial start, that equilibrium point may be stable. Okay.
Yes, sir.
But I have not seen that kind of case. Okay, for my models, whatever I have studied so far,
Yes, sir.
I found that at a time only one is stable.
Okay. Thank you, ma'am.
Thank you, madam, for the interesting lecture, and also thank you for mentioning our work. So due to lack of time, we'll move on to the next lecture. Thank you.
Yeah,
madam.
yeah, sure,
Thank
sir,
you.
sure,
Thank you,
sure.
madam.
Thairam, I deem it to be my pleasure to announce the next speaker, Professor Sandeep Banerjee. Dr. Sandeep Banerjee is a professor in the Department of Mathematics at the Indian Institute of Technology, Roorkee. He holds a PhD in Applied Mathematics from the University of Calcutta and has a postdoctoral fellowship from the University of Helsinki. With extensive teaching and research experience, Sir has published 44 international and 6 national papers, authored 4 international and 3 national books. Moreover, Sir received accolades such as the Indo-US Technology Forum Medal and Indo-US Fellowship. Sir has organized workshops, conferences, and seminars, contributed to course development, conducted sponsored projects, guided 7 PhD students, and delivered invited talks internationally. Today, Sir will be talking on the topic Effect of Productivity and Seasonal Variation on Phytoplankton Intermittency on a Micro Scale, an ecological study using closure approach. Over to Professor Sandeep Banerjee.
Thank you. Am I audible?
Sir, can you make your slides in the presentation mode, sir? It will be more
Yeah,
visible.
I'm just checking things actually. So am I audible?
Yes, sir, very much, sir, very much.
Okay, fine. And next is I have changed the topic actually. So now I will be talking about mathematical modeling of hepatitis C. So is it visible now?
Yes, sir, it's just loading. Is it full screen? Yes, sir, it's perfect now, sir. You are loud and
It's
clear.
okay.
Thank you.
Okay, I just want to check another thing. Just a minute. Whether you can hear the sound or not, just let me know. Is the sound audible?
No, means your voice is audible, sir. Are you playing any video, sir?
Okay, so can you see the video? No, you cannot.
No, no, sir. We can see the
We
slides
cannot.
and yourself.
Okay, just quickly. Can you now see?
We are able to see, sir, but we are not able to hear, sir.
Okay. Okay, we will skip that then. Oops. Okay, I think now the slides are visible.
No, sir. You need to share the screen, sir.
Okay, I think that is gone then. Oh, yeah, okay.
Yes, sir. Slides are visible.
Okay, now it is fine.
Yes.
Now quickly, can you see our video? This it appeared.
Video is there, but there's a kind of a delay, sir. We cannot hear the audio, sir.
Can you hear any sound?
No, sir.
Not the sound of the video. Okay, fine. Okay, so I thank Professor Vamsi for inviting me for the talk. So the title of my talk is Mathematical Modeling of Hepatitis C Virus. This one. So basically when you model any virus, you need to know a little bit about it. So basically it is a virus whose route is through the blood, and it as you can see that it is a virus which caused hepatitis C in humans, which means it's a cirrhosis of liver, some sort of liver cancer. Now this video, can you see the video?
Yes, sir.
Yeah, okay. So this video actually shows you that how a virus attacks a cell. So if you see that black dot which is approaching, So that black dot is some sort of virus. And what it does is it's sort of, fools your body, it fools your body's immune system to believe that it is a good thing that must need to enter your body. So this is most of the virus particle looks like, with a mechanism of lock and key. So the keys are on the body of the virus, and now it is find a suitable lock and make the lock believe that it's a good thing, and hence your body will allow this virus to get inside. So this is the kind of key and the lock, so known as cell receptor, and then it will allow the virus, it makes a passage, and the virus will go inside. So mostly this is how your virus enters your body. Okay, so why we are studying this? Because as you can see, the statistics shows that in America and worldwide there are 3.9 millions and 170 millions. In UK there are 4 lakhs and over 5 lakh infected in India. Now, what is the treatment available for this? So one is something called interferon alpha and another is called ribavirin. So this interferon alpha is already inside your body, and also it is synthetically available. And this ribavirin, it is a drug which is available. So a combined therapy or a monotherapy of interferon alpha is right now what is available in the market. There are also other drugs, but they are very, very costly. I mean, common people cannot afford that. Now, when this virus enters your body, it actually stays there, maybe in the latent form. So the the clinical name it is called sustained virological response, in short SVR, which means that, so when there is a virus inside your body, obviously you tend to take a blood test and see how many copies of the virus is present in the blood. So if it is 100 copies per ml of blood, you say, okay, you are clinically cured. But some remains of the virus is always inside the body, and ultimately this disease may result in the liver transplant, and if not, the patient dies. So how this works actually? So as I told you, interferon is already inside your body, and this virus, they tend to replicate. So interferon interfere in the replication of the virus and also helps to kill it. Along with, there is another drug which is called ribavirin, and you can see it is in the brand name of such and such that it is sold. And if you take both the drugs, there are 50% is the chance that you will be getting cured. Okay, so now how the virus actually, your immune system fights the virus. So if you can see that this virus, the way is always the blood. Now, what it does if you have seen that in the combat of jet planes, they throw some sort of flares, camouflage the enemy. Here also, the virus does the same thing. It throws a lot of the pink things that you get, and the immune system thinks that they are other entities, and they follow them, leaving the actual virus, which is, you can see in yellow. So they escape and then that's how the virus actually fools the immune system, and this is already seeing how it enters. Now what you see here is the different different phases that is observed in a patient suffering from hepatitis C. And the idea is to replicate this kind of behavior through the mathematical model. So this is now the hepatitis C virus, how it affects your liver. So what you see here is a healthy liver, and you will see soon that the virus enters the body through blood, either it is blood transfusion or something. So this virus, which is green in color, is a single-stranded RNA, and it is covered by these fatty envelopes to give it a protection. So it is entering through the bloodstream and sending some signals. It will locate where the liver cell is. So it will be attracted towards there. So here is the liver cell. It will get inside, release the virus replicates, one of them comes out, and again involved with the fatty acid infects the next cell. And that's how the process continues. So now let's come to the mathematical model. So we have taken these four equations. The first one is the healthy liver cells. This is the infected liver cells, infected virus, and non-infected virus. So the first one is a constant input. The second term is the growth. This is the death term, and this one I am coming. So this is one of the important parts. So what happens is this eta 1 and this eta r, they are the efficacies of the drug. Now when you take a drug, so not 100% of the drug works, a fraction of it works. So that's why it is multiplied by the C. So C eta 1 is the fraction of drug that works. And if you subtract it from 1, that means the non-efficacy of the drug, and which means your drug is not working. So due to the non-efficacy of the drug, the virus VI, it attacks the liver cell at a rate alpha and converts this whole thing to a infected liver cell. A similar thing happens here. This is now the combined efficacy of the drug eta 1 and eta r. Now you can ask that why you have taken eta 1 plus eta r by 2. Well, you could have taken in the form a eta 1 plus b eta r divided by a plus b. So it gives you almost the same results for all integer values of a and b. So why not keep the model simple? So that's why I have chosen a equal to b equal to 1. And that's why this is the form. So this is the combined efficacy of the drug, which results in making the infected virus non-infectious. And this is the failure of the efficacy, a combined effect of the efficacy of the drug. That's why 1 minus the combined effect, which makes the infectious virus to grow. So this is our mathematical model. Then you do all the necessary, mathematical testing of the model. One is finding the equilibrium point. So in this case, you have a disease-free equilibrium point and an endemic equilibrium point. And then you find whether your system is stable or not, and you find the condition for which your system is stable. And here the important thing is that we find something called eta C, which gives that if your eta is greater than some eta C, then you will have a viral load that your viral load will diminish and go to the SVR. That means you get rid of the virus clinically. And if it is less, then your infected steady state will remain there, but at some lower value. So basically you have two equilibrium points. One is virus free and one is endemic one, that is with virus. So we have the parameters from various journals. In this case, I couldn't get hold of any data from where I can estimate. So I have to rely on other published papers where you have to basically hunt for various parameter values. And once you do that, then you solve it numerically and explain the numerical results. So if you can see, so the interferon monotherapy, which means that only interferon is given to the body, and you can see that it has two phases. This is phase 1, a sharp decline, followed by a second phase. So in this case, there is no ribavirin, that's why your eta R is equal to 0. And you have eta 1, various efficacies of eta 1, that is interferon. And you see that you get cured sort of in approximately 23 days. And if you note that here it is 10 to the power 2, not 0, that is because clinically if you reach 10 to the power 2, it means that you are cure of hepatitis C. So that's why on the y-axis it has started with 10 to the power 2. Now, when this ribavirin is given, generally we are in the habit of having the drug that you take both the drugs at the same time, and it should work. Here also it started like that, but if you notice, of the ribavirin, and then there are varying values of the interferon. But here it comes to the same place at around 23. And if I compare both the figures, you don't find much difference, actually no difference between these two figures. Here the efficacy is 0, here with some efficacy. This is exactly what happened during the clinical trial. So what the doctors figured out is that if you just take the drug simultaneously, it really doesn't work. So what will happen is that you have to take interferon first, then after some days when the efficacy of the interferon goes down, at that particular point, you have to take this ribavirin, and only then it will work. So now we have fixed the efficacy of interferon to some lower value, like 0.4, and we vary the efficacy of the ribavirin, and you see that now with conjunction with the ribavirin, you can get cured in sort of 19 days, and the behavior is same as we have seen in the patient suffering from hepatitis C, that there is a first decline which is very sharp, followed by a second decline which is a little slower than the first one. Now, these are various cases which has been seen in patients in suffering from hepatitis C, that even if you give interferon monotherapy, it is not working. So it is the case of non-responders. So though it starts with a decline, but soon it goes to some steady state, non-zero steady state, where you are still suffering from hepatitis C virus. The same happens with the ribavirin also. They also tried with ribavirin monotherapy, but again, it didn't work. There is very less decline, but again ends with some endemic steady state value. Now, this is what happens when you stop the therapy in the middle. So as you can see that there is a first phase decline sharply, followed by a second phase. Then after 14 days, the 14th day, the drugs are stopped. So obviously, as expected, your virus will shoot up and it will go to some steady state value. Now, if you would like to generate this kind of curve, the trick is that this is not a continuous one. That will come out from the Mathematica or MATLAB which you will be using. So the trick is that you draw a curve up to 14 days, which will end here, and draw another curve whose initial condition will match exactly this point, and when you put your efficacy to be 0. Next curve will be generated from this point only, and then you just draw, just assemble both the curves. Because it has started from this point, obviously it will coincide with this point only. So this is one trick which can be used to generate the curve which you need. So this is another behavior which has also been noticed. It is called a triphasic decline, and now you understand that why it is called a triphasic, that because of first phase decline followed by a second phase decline and then followed by a third phase decline, and this is also getting a cure in a much less number of days. But this behavior is quite rare, though it has been observed in patients. Okay, so I will just quickly tell that why this model has been modified. So everything remains same, only you have added a proliferation term here, a logistic growth. This is due to the fact that in most of the patients, this kind of figure is seen, where you have a first phase decline and followed by a steady state, which is known as this shoulder phase, and then there is a second phase decline like this. So to capture that particular dynamics, we observe that if you add a proliferation term in the infected virus, like in this, then you will be able to get this kind of figure. So the conclusion is that, yeah, the idea was that whether the model which we propose is able to mimic most of the dynamics that is seen in patients suffering from hepatitis C virus or not, and we are able to capture most of the dynamics that is seen. So that was our aim, and it has been achieved. Now, once a model becomes a successful, you introduce delay, you introduce stochastic process and everything. But the take-home message is everything is fine, but as you can see, they give you the same kind of dynamics, only this kind of wavy nature came because of the delay that has been introduced to the system. So this will only fetch me some papers, but otherwise the first model is quite successful, and we are happy to keep that model that simple. So the only good thing came out from this delay model is that they have sort of matched with which the data which we have collected from this particular university associated with certain clinic. So that is one good thing only that came out from that particular study. So thank you. If you have any questions, I will be happy to answer.
Thank you, sir, for the very interesting talk. Actually, this is kind of work which I was looking, sir. We have worked qualitatively on the, here I have seen you are worked on the interferon, sir. We have worked on interleukin and also tumor nicotine factor for in the context of leprosy. So I was exactly looking this kind of work, sir. So I will surely keep in touch with you, and we can collaborate on the leprosy aspect, sir. Thank you so much, sir. Any quick questions from the participants? Okay, anyone from the Bharatwaj? No, okay. So let's thank sir. I mean, how many of you know Professor Sandeep Banerjee? I'm not sure, but all of us, the participants, know, he's a very well-known person in the field of mathematical biology, and sir is very friendly, and he's always coming forward to help all of us. Thank you. Thank you, sir, for taking your time.
Thank you. Thank you.
Thank you. So now we'll break for refreshments. 4:15, I request everyone to be here. We'll have a couple of paper presentations, and 4:45, we have the Mandir visit. So I request all the participants to leave their mobiles. In Mandir, we are not allowed mobiles and wallets, so you can just leave it here. It will be safe. The room will be locked. Once we come back from the Mandir, you can collect it. Okay, and I request all of them to wear ID cards. We'll be seated in a place. So when we are going to Mandir, please have your ID cards. Thank you.